Hello, everyone. Good afternoon. It's time, so I think we will get started. So this session is dedicated towards functional genomics, but before we start our session, we have a sponsor speaker from RNA Connect. So the first talk will be by George. That will be followed by our functional genomics session, which has Ralph from Memorial Sloan Kettering, Guru from University of Nebraska, and maybe myself. So we'll talk a little bit about our functional genomics score. So let's get started. George?
Thank you. Thank you. Hello, everybody. Thank you for being here. My name's George. I'm a research scientist for RNA Connect. This is our first time here at ABRF. We are very much having an excellent time here, meeting amazing people. This will not be our last time here. But I'm here today to talk to you about reverse transcription enzymes, both traditional RTs and their current limitations, as well as a new type of RT enzyme called ultramarathon, and some of the data that we've been getting when doing things like RNA-seq. So the majority of traditional RT enzymes are derived from retroviral RTs, which aren't very processive. They can copy about a couple of hundred base pairs at a time before falling off the template, then rebinding, falling off. So that's a couple of hundred base pairs, yet the average RNA length is around 2,000 base pairs. And we all know of many interesting RNA targets that are far longer than that. One of the main reasons for this lack of processivity is the inability for the RT to reverse transcribe through stable secondary RNA structures. Once it hits into a stable structure, it falls off the template. One way to address this limitation is these RTs have been engineered to be highly thermostable, meaning they can operate at high reaction temperatures. The idea being that these higher reaction temperatures will help destabilize the secondary structure and allow for the RT enzyme to be a bit more processive. But it's not enough heat to denature the full structure, so you're actually not achieving your first goal. And higher temperatures actually degrade RNA, so you're destroying the thing that you want to study. As you can imagine, this is a very large problem. As RNA researchers, we can't detect all of the RNA that's in a given sample. We can only analyze a subset of it. If it's highly abundant, short, or unstructured, no problem. But once you start to probe the more complex RNA, like really long, highly structured, modifications, different isoforms, you start to hit into the limitations that these RT enzymes have. So the real question is, what are we missing? How deep is the iceberg beneath the surface? Maybe our understanding of RNA function is limited because the functional characteristics of the RNA is currently invisible to us. What if we can solve this problem by using a fundamentally different type of RT enzyme? And that's exactly what ultramarathon is. It is a group 2 intron-encoded RT, so it's structurally different than a retroviral RT. And these differences in structure allow it to be extremely processive. We've seen reads longer than 30 kb when we do long-read sequencing. And one major reason why it's so processive is because it has helicase activity. It can easily unwind these stable RNA structures without the need to heat up the reaction temperature. So you can actually perform RT at 30 degrees, even room temp, if needed. It also has template switching activity, so you can add essentially unique barcodes at the 5' and 3' ends of all of your RNA transcripts and perform things like RNA-seq. There are many different applications. If you're interested in RNA structure profiling or natural RNA modifications, we have a number of publications on that. I suggest you check it out. But in the interest of time, I'm only going to be going over 2 main applications: RNA-seq library preparation and RT-qPCR. So we performed RNA-seq on human lung RNA using ultramarathon RT, or UMRT for short. And we compared it to 2 widely used library prep kits that contain traditional RT enzymes, one from Takara and one from NEB. And we found that when using UMRT, we actually get just about double the genome coverage across all 24 human chromosomes. And this is interesting because right now it's estimated that around 2% of the human genome is devoted to protein coding genes. And then another 5 to 7 to 10, depending on who you ask, is long non-coding RNAs that have at least been identified or reported in literature. Those estimates are based off of data that was obtained by using traditional RT enzymes. And the results that we found does agree with that. However, when using UMRT, we're getting genome coverage 15%, 20% genome coverage, even more. So what is UMRT seeing that these other RTs are not? We can start by looking at read distribution. If we take the total number of reads and partition them into their respective RNA gene features, we can see that traditional RT enzymes, now in this case, Takara and NEB have very similar results, so I'm just showing one, but they use around 80% of their total reads to identify genes, where UMRT only uses about 50%, about half of its reads, to identify genes, which means the other half can be used to discover new biology. For instance, around 18% of our reads are unannotated. And this also makes sense because those annotations were derived from data obtained by traditional RT enzymes. So because they aren't as processive, there will be a lot of unannotation. So this is a whole new area of functional discovery that's waiting. Another interesting thing is that we can identify a lot of introns. And this is kind of interesting because we're using oligo-DT, so why are we seeing so many introns? So we continued our analysis, and we can see that despite the fact that UMRT only uses half of its reads to identify genes, we can still identify more genes than traditional RTs. And we've seen this using different types of RNA from different organisms. We've compared to the superscripts. We always identify more genes, but it's not like we're identifying double the amount of genes. So if you dig a little bit deeper, we observe that UMRT can actually detect around 3 times more retained introns than other RTs. And these are essentially an RNA isoform where the intron doesn't get spliced out and it stays in the full RNA transcript. And this actually makes a lot of sense because introns generally are highly structured. They have to fold in on themselves to bring together 2 exons during splicing. And some of these exons can be extremely long. This is just one example. We can see that there is a really long intron of tens of thousands of base pairs long that UMRT gets really amazing read counts, really good coverage across this really long intron range, something that's invisible to existing RTs. Remember the iceberg idea? We're starting to see what's below the surface. There are 2 main reasons why someone would do RNA-seq. One is to identify genes. The next is to quantify genes. So when using UMRT to perform RNA-seq, our gene quantification is extremely reproducible. There's very low sample-to-sample variation, as you see in the log scale TPM in the top one of the 2 figures here. But interestingly, when you compare UMRT to traditional RT enzymes in the figure below, you can see that, we found that traditional RTs tend to underestimate the gene abundance of a large subset of RNAs. And when looking into this a bit, we see that they were actually highly structured. And this makes sense. If the existing RT enzyme has a hard time or isn't very efficient at reverse transcribing these difficult, highly structured templates, there will be lower read counts for them, therefore lower gene abundances. So we've actually validated a subset of these using both our own RT-qPCR kit as well as an NEB one-step universal RT-PCR kit and found that both of those kits agree with UMRT's RNA-seq gene abundance. So the final slide I have is going over RT-qPCR applications. Now, we've all had the joyous, wonderful experience of designing and optimizing RT-qPCR primers. Step 1, have software generate about 10 different primer pairs that are located in different regions of the target transcript. Step 2, test all of those primer pairs. Step 3, believe in your heart that the gene abundance that is given to you by that primer pair is true. Well, the good news is that if you use UMRT to perform RT-qPCR, your life just got way easier because UMRT provides full 5 to 3N full transcript length. So now it no longer matters where your amplicon is. For instance, here in this experiment, we designed 10 different primer pairs that were evenly spaced across a 7 KB transcript. This particular transcript had varying amounts of GC content, some as high as 75%. And regardless of where the amplicon's located or what the GC content is, UMRT provides very consistent, almost identical CT values you see here in the dark blue bars. However, when using other RTs, the CT values are highly variable depending on where that amplicon is. So the point is that you never know when your gene quantification is biased by the RTs that you use. That's all I have for you today. I want to thank you for your time. Here are 4 kits that we currently have available. We have many more coming soon. We have full library prep kits coming out. We'll have long-read library prep kits on the way out as well. There isn't that much time left in the booth area, but if you have any questions at all, please feel free to stop by booth 509, and I'd be happy to discuss whatever kind of projects you have. And next, enjoy these amazing talks from these amazing people.
Thank you, George. So in the interest of time and the fact that we have a very packed session, we are going to hold off the questions till the end of the session. Okay, so next speaker is Ralph Garippa from Memorial Sloan Kettering's Genome Editing and Screening Core.
So 2 years ago in Boston, Molly and I were talking about how good the talks were, but really mostly about HiSeq, about FACSFlow, about proteomics, and we felt that functional genomics needed to have its place, so we put in an abstract, and this is the fruition of that abstract. So the reason we wanted to talk about functional genomics is that you might have a core, a small core, you might be thinking about starting a core, or maybe you don't have an appreciation of the power of what a functional genomics core can do for you. So we wanted to tell you how to start it up and how to get it ready for prime time. So in my core facility at Memorial Sloan Kettering in New York City, we really focus on CRISPR and RNAi-based capabilities. Number one, we have molecular biology. We can make you individual hairpins or guides for CRISPR. We do plasmid vector modification. We design and clone custom libraries based on your gene of interest list. We can do genomic DNA extraction, HiSeq sample prep. We make barcode libraries because sometimes you just want to trace, do lineage tracing without perturbation, and see DNA cloning. In the second module, we have a cell line service. We can knock in or knock out any gene of interest. And we've started in the last year doing a lot with base editing and prime editing. One of the reasons why is if you want to do saturation mutagenesis of a very defined area, you can do it better with prime editing because you have more sites at your disposal. You're not limited to the PAM sites. We do virus production as well as concentrated virus because there are some cell types like T cells and macrophages that won't take up virus readily. And then we also have a screening service. We can do whole genome pooled CRISPR. The penultimate libraries out there now are wine and cheese, Brunello and Brie, human and mouse. We can do pooled-based screen deconvolution of your HiSeq. We use MAGIC and other platforms for analysis. We've brought in CRISPR-A technology for amplification of gene, CRISPR-I for blocking, the counterpart to RNAi. And we also have a full genome collection of siRNA if you want to do well-by-well phenotypic comparison of what is going on with your gene knockdown. So in the early days, all we heard about was off-target effects. So we decided to do something about it and create a better algorithm. So my core partnered with Christina Leslie, Andrea Ventura, and Scott Lowe, and we published this new algorithm called GuideScan. And GuideScan really was superior to the other 3 most popular ones on the market. If you look at the bottom of the screen, GuideScan is on the far right with the black bars. The other three are to the left. And you see the red and the white regions. That actually shows you the number of sequences that would target off-target. So ours was very, very clean. And if you look down below, you actually see the number of off-target effects, and it pretty much goes to zero in the far right, whereas the other pre-existing programs had a lot more propensity. This was very popular. And in fact, Sloan Kettering gets requests by private pharma to license this so they could do their own designs. So we came out a couple years later with GuideScan 2. This has higher specificity, it has higher cutting efficiency for both human and mouse, and importantly, you can apply it to CRISPR-A and CRISPR-I libraries. Now, the problem of off-target wasn't only to CRISPR, it was also for shRNA, so we made a better tool and published that. So again, working with Rafi Pelisoff and Christina Leslie, we published this tool. It's a sequential classifier that allows us to make better shRNA with fewer off-target effects. So if you look in the middle here, the text says, outperforms other algorithms 90%. Of the predictions trigger 85% knockdown. So we really are close. Even though Fire and Mello won the Nobel Prize 25 years ago for the discovery of RNAi, people have not stopped improving the technology. And today, even if you think about it, a CRISPR, some of those knockouts, those indels will be in frame and you'll get a very small effect. Now with shRNA triggering knockdown well over 85%, you can almost get equivalent results. So that really solved our problem with the algorithms for designing CRISPR and RNAi. And then we started to look at the backbone. So starting with the early PLKO backbones that worked very well in the TRC collection, we migrated to microRNA 30, and then finally to the high-efficiency MIR-E. MIR-E has a conserved motif, and that's very critical for biogenesis. So in the bottom left, you can see the MIR-30. About 50% of our designs would prove a very high knockdown. But now putting those same sequences into a different backbone that has better biogenesis, actually both work very, very well on the Western blot. So you can get single copy effective shRNA. Now we needed a way to bring these improved SH and sgRNAs into cells, so we expanded our war chest of plasmid vectors. So any investigator that comes to us now can have a constitutive or an inducible, a green or a red, a puroselective or a neoselective, and almost anything else. If you need BLAST or ZEO, we can clone it in and customize that vector for your experiment of interest. So what is pooled screening? And I'm going to take a minute to talk about it because honestly, when I came to this field 13 years ago, I didn't know much about it. So for pooled screening, you have to start with a concept for your biology. What is the separator going to be that your perturbation is going to reveal to you? And I'll have another slide on that. So what are you screening for? Are you really looking for a depletion of the guides in there or an enrichment of the guides? And that'll all show up in your HiSeq readout. Is it drug sensitivity? Is it drug resistance? Is it expression of a reporter or a metastatic event? And then you might wonder, should I use CRISPR or RNAi? But remember, CRISPR, even though it's more popular now, uses a bacterial endonuclease. RNAi is a natural process in all mammalian cells. So some cells that don't adapt to CRISPR will adapt very well to RNAi. And for CRISPR, you have to consider using a single vector or a two-vector system, right? And efficiency is the trade-off. If you're going to load that vector with Cas9 plus the guide plus any other features, you will lose about a log of efficiency. So you have to factor that in. Most of our pooled screens, we start with a stable Cas9, and then we bring in the library. We also have full collection SI, as I mentioned earlier. This gives very nice knockdown within a 3 to 4 day window. If you need a more durable event, you can't use it. So to run the screen, you package your virus, you infect, you transduce the guides. And then the guide representation, if you're going to have a dropout screen, you better stay high, about 1,000 representation. And for a positive selection screen, usually 300 to 500 is fine. And technical replicates should be between 3 and 4. These are examples of pooled screens. So if you look at it, we package the in lenti, either the guide or the hairpin. We infect those cells, and then this is the separator. So on top, for a viability screen, if I knock out a tumor suppressor gene, you're probably going to get an increase in cell. In enrichment, if I knock out an oncogene, you will get a depletion in that population, in simplest terms. Or you might have a reporter, reporter that's enriched or depleted, and you can figure that out in a fax flow system, or a behavioral screen, such as invasion and metastasis. So I'll walk you around the horn. We'll start at 12 o'clock here. Many investigators come to us with a gene list. We then use either Splash or Guide Scan to do all the designs. We've been using Agilent over years because they have very tight quality control. And in 12 years we've been doing this, we only had to send one chip back that they replaced for free. We put it into our vector on the bottom, and then transduce and select. Now, at this point, you could add a drug or withdraw a drug. You could add radiation if you're looking for a radiation sensitizer, run the screen, and then finally get your readout on HiSeq. So we always recommend using a multiplicity of infection, pretty low at around 0.3. Why? Well, if you look at the third bar in this sequence, you'll see that you'll get about 85% of your cells will only have one guide. So you will get an unambiguous readout in your HiSeq if you keep your MOI pretty low. Otherwise, you're going to be reading 2, maybe 3 guides in the same cell. And this is what the expression looks like. So on the baseline, you see that's your cells. You'll infect them. So now you see a second population showing in. In this case, we did a selection under neomycin, and there's an enrichment in the upper left. And then finally, we added docs for the inducibility, and you see your population now carrying all the hairpins that you want. You bring that forward in your screen. So here's a couple of resources I put in, and I know these slides will be published, but after we did this for a couple of years, we published these 2 papers. The first one is how to do an in vitro screen. The second one, how to do an in vivo screen. And by this point, we had developed our algorithms, we had our backbones in place, we had published, and now we turned ourselves loose on the rest of the faculty. So I'll give you 2 examples of pooled screens, and I chose these 2 because the questions arose at the bedside, and then we brought those questions back to the bench, figured them out, and then brought them back to the bedside for the patients. So here are 2 examples. So the first one is looking at an enzyme called protein arginine methyltransferase 5, or PRMT5. It is an attractive target in lymphoma. The reason is that it's essential for B-cell lymphoma survival. And so a full CRISPR screen was done. You see that in the bottom left. And in fact, it was done in 2 different cell types, in Z138 in the OCH cells. And across those 2 cell lines, there was a very small number of hits, including PRMT5. And when we carried that out into other cell lines from B-cell lymphoma, in fact, we saw that it was absolutely essential. So now we know PRMT5 is a drug target. Drugs were developed, and those are coming up next. You see one of them here on the right side. This is a GSK compound, 591. But really, 3 different selective inhibitors went into the clinic at the same time for blood cancer patients. And like any other drug modality, you know the resistance is coming. So the investigators, I thought, very smartly came to us and said, where is this cancer going to go? How is it going to get around our PRMT5 inhibition? Can you do a CRISPR screen and tell us what the most likely avenue of escape is? So that's what we did for them. So we used the Brunello full genome CRISPR library, 77,000 guides. This is a lot of cell culture. So about the top half of an incubator will be dedicated for like 10 to 14 days if you're running this screen. So we infected, we transduced, and you can see in the center, we either had a DMSO control or the GSK 591 in the drug treated. And the results of the screen are on the far right, and it was unmistakable that P53 was the top ranked sensitizer of B-cell lymphoma. The driver of resistance was, in fact, Musashi 2. So just to confirm our results, we took, instead of the 77,000 guides, we made a smaller library of all the hits, so 405 genes with about 5 guides per gene, ran the same screen with drug and without drug, and unmistakably reconfirmed that P53 was the top sensitizer and driver of resistance was going to be Musashi. So what did that mean? It meant that we discovered a therapeutic strategy that should be implemented along with PRMT5 inhibition, is to bring in either Musashi inhibition or BCL2 inhibition to block the most likely avenues of escape by the cells that are being treated. So the last example that I'll show you of pooled screening is coming up. So many of us last November read this article in Nature about a virologist who had breast cancer. Her name is Beata Halassi, and her breast cancer was not responding to conventional treatment, so she took matters in her own hands. Being a virologist, she created oncoviruses in her lab, treated herself with that QC'd material, and in fact, the breast cancer shrunk. It softened to the point that it could be removed surgically. She had a lot of trouble publishing this work. The 7th Journal finally picked it up. Not that she was supporting treating ourselves for cancer, but rather pointing out the importance of utilization of oncoviruses. So we're not using the particular virus that Beata had worked with, and there is only one oncolytic virus on the market that's called Imlygic. It's TVEC. It's approved for melanoma, but there's plenty of others that are in phase 1, 2, or 3 trials. 3 are listed at the bottom here for glioma and brain cancer. So how do you get an oncolytic virus approved and on the market? Well, the one that one of our staff members, Charlie Rudin, is working with is called Seneca Valley virus. It infects specifically cancers that have neuroendocrine features. It's a picornidae virus. It's an RNA virus. And if you look in the bottom left, you'll see that for small cell lung cancer, quite a few of those lung cancers are infected by this virus. Not so many non-small cell lung cancers in the center, but almost every pediatric cancer that we came across. And this group of patients, by the way, 30,000 die every year. So this is a big case to be able to solve it. So if you look at the life cycle of the Seneca Valley virus, when it infects cancer cells, you see in the upper left there's receptor binding. It internalizes, and like many other viruses, it takes over the protein synthetic machinery. So it hogs all of the transcription and translation to the point of making more of itself until the point where the cell has to burst. So that is the lytic feature of the oncolytic virus. So before I change the slide, I want you to look back in the upper left at the receptor binding, because I'm going to circle back to it. So could you really make Seneca Valley virus into a drug? Well, number one, if you give it IV, it doesn't integrate with the host genome. It has a replication competent life cycle, about 8 hours, and it persists in the human body for about a month. It has no DNA stage. It's immunologically stable. And it's not inactivated by blood components, nor does it cause hemagglutination. Beautifully, it homes to the tumor. No matter where you inject it, pre-existing antibodies are low. And the fact that always blows me away is it has greater than 10,000-fold selectivity for the tumor over normal human tissues, and not known on its own to cause disease. So these are very favorable characteristics that suggest it could be a drug. And in fact, when John Poirier and Charlie Rudin injected this into mice, this is looking at immunocompromised hind flank pockets. And you could see in the bottom, the GFP is showing up very well. On the top, for two different preparations, the LX22 and the LX36, you see profound tumor volume shrinkage. And when they went to the higher dose, it was a flat line. There was no tumor at all. So again, very compelling evidence in animals. So now they went into a phase 1 trial, and the injection on the left is, you can see the increase in the SVV as the bar goes up. You start to see neutralizing antibodies in the red about 7 days later. And then let's put our attention over on the blue bars on the right. So these are the patients in the phase 1 trial. So a number of them here had very encouraging results: tumor shrinkage, marker decline, some very positive results. One of the patients in the black bar passed away during the trial, but others had no effect at all. So the question is, how do you recruit patients for the phase 2? How do you select the ones that are going to have blue bar effects? So we didn't know. So we thought, well, the first place to start is to find that receptor. What is that receptor? And maybe we could use it as a biomarker. So CRISPR had just come out around that time, and all their previous attempts to find that receptor failed. They tried RNAi screens, they tried cDNA add-back, they tried cell surface glycoprotein capture. None of them worked. Why didn't they work? Well, it could be low abundance of the receptor protein or incomplete knockdown, right? Because if you have even one receptor remaining on the surface, the virus will get in and start the life cycle. Maybe there's a complex of proteins or multiple receptors. Maybe you need more than one factor for permissivity. And so just kind of a little spoiler, I put an asterisk next to the ones that I'm going to show you turned out to be true. So we did a genome-wide lenti screen. The genomic knockout library, otherwise known as GECKO, CRISPR knockout library, was the penultimate library at the time. And we were using the pathway on the far left, which is non-homologous end joining, to induce indels, either deletions or indels involving additions that would throw the gene sequence out and create a knockout. So I thought it was very smart working with John Poirier and Lindy Miles. This was the centerpiece of her PhD. They chose two different cell lines, one that was very permissive for the virus infection and one that was much more resistant and had low permissivity. So the high one was HL446 and the low one was HAP1. And then we did a positive selection screen, meaning that the only cells remaining will be the ones that knocked out the protein that the receptor, that had the receptor that the virus would attach to. So here's the workings of the screen. We optimized it for the number of cells, for the puromycin selectivity. One cell grows faster than the other. The point of this slide is that we didn't do it one size fit all. We optimized it for each cell line. So this is the results as they came back from the HiSeq, and we were just blown away. You couldn't miss the fact that the ANTXR1 protein was the receptor for the Seneca Valley virus, both in the HAP1 and in the 446 cells. Now, just to confirm this, and you see some other passenger genes that are coming along, but the percentage of colonies for ANTXR1 was screamingly high. So now we did a secondary screen taking individual guides targeting ANTXR1, and in fact, we concluded that it certainly was ANTXR1. What is that protein? So it's a single transmembrane receptor. Importantly, previously, it had been identified as tumor endothelial marker 8, or TEM8, which is overexpressed in tumor endothelial cells. Now, opportunistically, anthrax, bacillus, not virus, but toxin, uses the same receptor to get in the cells. So this is a convergent evolution where a virus and a bacterial toxin use the same receptor to get into a cell. Importantly, there's two different receptors for anthrax, anthrax 1 and anthrax 2, and that'll show up important in a minute. So what we did now is look at a number of different insertions and deletions, and you could see those in the upper left in panel A. So if you look at number 4, number 12, number 20, you see that there's insertions or deletions. In B panel and in the lower left, you see flat line. None of the cells die when you knock out and you have a really productive indel. The rest of them will die. And then looking over on the right, you see different cell lines that we're able to prove this. And the far right, that black bar that's up high, that's when we added back the ANTXR1 receptor to cells that already had it knocked out. And looking further into the re-expression of the protein, you can see that the knockout's very effective. We created a GFP Seneca Valley virus, and microscopically, as well as by the Western blot at the bottom using an H8 tag, we were able to show that, in fact, it was the ANTXR1 protein. Now, if you look at the small box that's in the center there under permissive, you see 20 and 0 for ANTXR1 plus and minus. So that means if a cell or a patient's going to respond to Seneca Valley virus, they have to have ANTXR1. But if you look over to the non-permissive, you see there are a number of other cells that have the Seneca Valley virus receptor but aren't susceptible. So why is that? There has to be a second factor. So what we did is we looked at the Cancer Cell Line Encyclopedia, which has over 1,000 different cancer lines, and the results for that are in panel B and panel C. And what jumps out at you in the upper right in the blue bars is interferon alpha and interferon gamma. So yes, if a cell's permissive, it has to have the anthrax receptor, or TM8, but it also has to have defects in innate immune signaling. That's the key. That's how you field patients for the second trial to run. And just to wrap this up, if you look in panel A in the upper left, this is adding back a reporter. We create an FC chimera reporter, and the more of the receptor you add back, the more binding you get. The binding is not displaceable by high levels of sodium chloride in the middle panel. And then on C, on the bottom, this is adding back ANTXR2 receptor, and you get no binding at all. So the bacterial toxin will bind to it, but the Seneca Valley virus is very selective for the TEM8 or the ANTXR1. And then why is this important? Well, if you look at neuroendocrine tumors, they're heterogeneous like most tumors. 7 different types of cells within that tumor, including cancer stem cells, endothelial cells, parasites, cancer-associated fibroblasts, are all killed, are all lysed by the Seneca Valley virus. So what was originally positioned as a single modality for treating neuroendocrine cancers now is being repositioned to be able to take cold tumors and make them hot. Okay, so now you don't need the high dose anymore, you don't have to lyse the entire tumor, but if you can take an administration of Seneca Valley virus, make that cold tumor hot, now you can come back with other cellular modalities like CAR T and really have an effect. So I've just put here 3 different recent publications to convince you, hopefully, that not only oncolytic virus as a modality together with cell therapy, but also TEM as a bona fide biomarker of neuroendocrine cancers is quite relevant. And with that, I'll show you my group. So this is the Magnificent Seven. This is Jesse and Claire, Sanjay, Annie. Our admin is Anna's here, as well as our lab manager, John Min. Hannah's down in the lower left. So this group did all of the work I showed you, as well as many other projects I didn't have time for. So thank you for your time. That's functional genomics as we do it.
Okay, so my name is Guru. I'm going to switch gears towards studying one gene at a time, which is like making one knockout mouse or one knock-in mouse, like studying function of single genes. So I'm going to talk about newer technologies for editing the mouse genome. My name is Guru. I'm from University of Nebraska Medical Center. In a few days, I'll be changing my university. It's going to be University of Mississippi Medical Center. So I'm going to talk about 2 things. One is like our previous technologies and newer technologies that are coming and that are in the pipeline. So first, the previous technologies are Easy CRISPR and Gonad. I'm sure some of you may have heard about it. Please ignore these first 5 minutes. So Easy CRISPR. What are the types of genome edits that we do in the mice? There are 4 categories. One is knockout. Second one is small changes. Third one is large cassette insertion. And fourth one is gene replacement. And the first two categories, usually we get about 20% of the times in a transgenic core research, like requests. And large cassettes and gene replacement is about 80% of the projects. But the first two are very easy to do, and many core facilities easily adopted CRISPR technology within a couple of years to generate knockout and small changes. But large cassettes and the last two categories is quite hard. And one classical example for gene replacement is conditional knockout. And we solved this problem in 2016, almost 10 years ago, by developing a method called Easy CRISPR, which stands for efficient additions with ssDNA inserts CRISPR. The difference between the previous methods that people were trying to put large cassettes is plasmid DNA, whereas we used long single-stranded DNA that really changed the outcome. And we published this paper in 2017, and which is already downloaded, like accessed more than 50,000 times, and it is cited several hundred times. And we published this Easy CRISPR protocol paper in 2018. And just to summarize Easy CRISPR, if you want to create a knock-in mouse, for example, GFP into the N-terminus of your gene, all you need to do is that find a guide RNA that cuts close to the ATG and make a single-stranded DNA with just 50 bases homology arm on the left, 50 bases homology arm on the right, and insert that cassette. And if you want to change that to the C-terminus, all you need to do is that change the guide and change the homology arms, cassette remains the same. And if you want to create a conditional knockout, bottom is the conditional knockout and the top is the wild type allele. There are two LOXP sites that are inserted to get to the bottom cassette. What you do is that, like, you take two LOXP sites, stitch them together in long single-stranded DNA, make two cuts in the genome, drop the cassette in, and insert the new cassette inside. It's as easy as that. And in some genes, it is nearly 100% efficient. Just like 3-4 pups born, and all of them have conditional knockout cassette. Second method is GONAD, which is like in vivo genome editing method. So left side is a traditional method where you take like 10 to 15 male mice and 10 to 15 female mice and isolate, made them together. Next day morning, you euthanize the females, isolate the zygotes, inject one by one, and transfer them into next set of females, and wait for pups to be born. This is the traditional method. Instead of doing all these cumbersome methods of 3 steps, we thought, why not use the same female that was mated with a boy and do in vivo genome editing? What we did is that we took this mice and then did the surgery and, oops. I don't know, there is no pointer here? Okay. No pointer. Okay, it's okay. So we did the whole process in the mouse, and this is the proof of principle experiment on the right to show that like GFP mRNA and GFP guides can be delivered. And we gave this method a name called GONAD, which stands for Genome Editing via Oviductal Nucleic Acids Delivery Method, which is a microinjection independent genome editing method. And here is how the working protocol is. That like, left side is a mouse that is anesthetized, she's pregnant, and you surgically open her oviduct and inject CRISPR-Cas solution into the oviduct. There are tiny round things that you see are zygotes. And then on the right side top panel, what you see is that the stretched out zygote, like a oviduct, and on top, we hold it with 2 electrodes and then apply electroporation. And after that, we suture the female back, left side bottom. She's awake after surgery, and then after 3 weeks, she will develop her pups. And I'm going to show a small video on this one. Like, you will have to click start there. Okay, so this is the oviduct region that is imaged under microscope. We are injecting approximately about 1 microliter solution into the oviduct. You can see it is swelling. Inside this oviduct are the fertilized zygotes. They are approximately about 10 to 15 hours old, and they are single cell stage. And they are present inside a thick cumulus cells. They are very tightly packed. And after we let the animal stay for about a minute or a couple minutes, so that like all the solution gets dispersed within the cumulus cells. And then after that, we position the mice and take Kim wipe, that is a tissue paper that is soaked in PBS to keep the tissue moist by placing on top of it. And after that, we grab the tissue using custom electrodes that are approximately just 2 to 3 millimeter in diameter. And once after we grab that, we just press the button to apply electroporation. You can just watch for a few seconds now here. you will see air bubbles, and that is the stage when all the magic happens. So that's when all the CRISPR-Cas solutions enter into the zygotes that are present inside the oviduct, and then we suture this animal, and she will develop her own pups after 3 weeks. So we published this in Nature Protocols in 2019, and we published a paper in, again, 2019 in Genome Biology. So I'm going to talk in next few minutes about the limitations of these technologies and what we are developing to solve some of these. So major limitations of EasyCRISPR and GONAD is size of the insertion cassette. Up to 2 kb, they work beautifully well, particularly EasyCRISPR, and GONAD doesn't work more than 1 kb. And the insertions are not very perfect. Like sometimes there will be misincorporations because we provide only one strand. The other strand is synthesized by the cellular repair system, and it is not accurate all the time. So you will see either point mutations or sometimes imperfect, like partial insertions of the cassettes. So we have right now developing 3 technologies in the laboratory. One is called re-engineering, second one is script, and the third one is T4 artificial viral vectors. I'm going to talk about re-engineering, which actually I changed that name to recreating, which I'm going to talk to you in a minute. So I'm sure many of you have heard about a classical Rosa knock-in mouse called MTNG mouse that has like a TD tomato that is a red-colored fluorescent protein that is driven by CAG promoter, and which is floxed. Floxed means it has 2 loxP and once you breed this mouse with a Cree, the GFP gene expresses. It is like a traffic light reporter mouse. What we did is that instead of, people ask us to generate large knock-ins into Rosa locus. What we did is that instead of using a wild-type mouse with a lot of plasmid big constructs, we took this mouse and then we inserted just a guide RNA, we cut a guide RNA and insert cDNA of interest, that is a blue-colored rectangle that you see. And the idea is that, like in this mouse, you will express cDNA of interest ubiquitously throughout the mouse. And the second idea is that, like, we found a guide RNA that cuts upstream of GFP, and if you insert there, after breeding the Cree-lock system, at the bottom, you will see that, like, your cDNA is expressed only in the Cree-lock system. And the other possibilities of re-engineering, we call it as like, if you want to convert an existing mouse that does not have any lock-stop locks, if it is a constitutive expression, you can convert into conditional expression by inserting lock-stop locks. Second one is like if you want to insert a fluorescent protein into your existing Cree mice, for example, like you will create a GFP knock-in, like a GFP reporter mouse. And the third one is a very classical functional experiment where if you have multiple different isoforms of the same gene, and if you want to express them at an equimolar concentration, what you do is that you create a mouse A, and then after you create a mouse A, you take the A out and put gene B, that is isoform B, and then you can use them as trans heterozygotes to express both copies at the same concentration. So I can talk more about this later, like if you have any questions, but some of these concepts we have already proven. And right now we have developed more than 15 mouse models using all these technologies, and then we are submitting this manuscript very soon. And I gave this name, re-engineering, to recreating. Recreating stands for re-engineering with CRISPR of previously engineered alleles to insert new genes. That means like we are using these existing cassettes, what I told you, like there are 3 examples. We use the existing mice and recreate those mice. So recreating of AI95, this is another example of a different Rosa locus. AI stands for Allen Institute. Some of these mouse models are very popular. They are in Jackson Laboratory, and you can use these mice. And then what we do is that, like, instead of use the existing cassette, we insert a new cassette by recreating, and so these mouse models are shown to be working all the time, and the new cassette that you insert will almost always work. So, and this is another classical example where you can use 2 different cassettes. One is constitutive expression, another one is tetracycline expression. Both can be achieved by creating a single mouse. And there are no equivalent MTMG mouse that is like traffic, like MTMG like rat, that is like a traffic light reporter kind of rat. So we created a smart rat, which stands for scarlet red flanked with multiple array recombinase sites turning green. So the idea is that we took the M scarlet red and flanked with multiple recombinases like WOX, FRT, ROX, FLOX. And then we inserted this into the rosa locus. And after you breed with any of these rats, you will be able to convert red into green. And we call this as smart rat. And currently, this as soon as I made this rat, I deposited to Rat Regional Resource Center that is in Missouri. And anybody who is interested in working on rat model can procure this mouse rat. So second technology is script. And so script is stands for simultaneous CRISPR and PITT. I'm going to just tell you a little bit about PITT. That was a technology developed about 15 years ago now. And so left side is a DNA piece. If you want to introduce into mouse, what we do is that we inject into zygotes. Once you inject into zygotes, that DNA can go randomly into either 4th chromosome or 11th chromosome or somewhere. You don't have any control on that. So if you want to control it exactly where you want it, instead of going into euchromatic region, which is going to be dead, if you want to put it into only euchromatic region, we did a landing pad. We created a landing pad in the mouse genome by putting 2 different kind of heterotypic lock-speed sites. One is red colored, another one is orange colored, and that we call it as landing pad with a neomycin cassette. And then we go with the donor DNA along with the CRE with the heterotypic lock-speed sites. What it does is that the DOI, that is donor DNA of interest, gets inserted into the rosa locus, and this is creating 2 steps. So this was published 15 years ago, and what I thought is that instead of creating that landing pad using embryonic stem cell method, we can do both things simultaneously. The idea is that there are 2 reactions. One is green on the left, and one at the bottom is the gray, you can see. In the first reaction, so what happens is that Cas9, which is blue colored molecule, and guide RNA, which is green, and then the ATP site, it inserts the small landing pad. And in the same microinjection mix, we provide PITT components, which has integrase mRNA on the right side gray rectangle that you see. And there is also donor DNA of interest. And once that ATP site is inserted in the reaction 2, that integrase mRNA gets produced into integrase protein, and protein picks up that donor DNA and inserted the cut site. Okay, so this is one of the proof of principle experiment showing about 15 kb knock-in mouse was created. And then the last one is T4 AVVs, which is still very primitive. I'm going to skip these slides because I want to give opportunity for Molly to speak. Okay, I'm going to stop here. Okay, you can stop. Thanks.
Thank you, Ralph and Guru. Those were amazing talks, and I don't think my talk comes anywhere close to it. I'm a relatively small core, and my core is similar to what Ralph does, but we are sort of in our infancy. I look up to Ralph to hopefully one day make my core more like his, where we are doing some novel stuff. So I am Molishree Joshi. I usually go by Molly. I'm the Associate Director for the Functional Genomics Core for University of Colorado. We are housed on the Anschutz campus, but we are a core for all academic institutes in Colorado. I'm also the co-chair for the Genome Editing Research Group of ABRF. So if any of you are working in the field of genome editing, functional genomics, shRNA, and would like to join our group, we are recruiting. Please come talk to me after this session. Okay, so my talk is going to be slightly different in the sense I'm going to talk about our journey, what brought us here, and what direction we are going. So this is a busy slide, but it kind of is our history, how we started. So the Functional Genomics Shared Resource started on the CU Boulder campus by the collaboration of Espinosa Lab, University of Colorado's Cancer Center, BioFrontiers Institute, and Molecular and Cellular MCDB department. So essentially in 2010, the PIs of Colorado really wanted to do shRNA screens, and buying libraries from companies was too expensive, and so the idea was, okay, we should have our own core. And so we basically bought the TRC shRNA library for human and mouse, and that started the functional genomics core. I joined the core in 2013, July of 2013, and then the CRISPR revolution started end of 2013, beginning of 2014, and I'm so glad I was there to go with the flow. So over the years, we've expanded our core. We have the TRC libraries for gene loss of gene function studies. Then we added the ORF libraries, which are used for gene upregulation. In 2013, when CRISPR revolution happened and a CRISPR arrayed library was available, we acquired that. But even before that, I was tasked with teaching myself everything about CRISPR. I learned how to clone guide RNA. I learned how to amplify CRISPR libraries, the Gecko library, the Brunello library. I established all those protocols on campus. Then I taught myself how to edit genes in a variety of cells. And I also applied for a bunch of internal and external small grants to help my core do some automation. So, okay, so we've always been a very, very small core. It's either been a 1 to 3 FTE. So right now it's just me and Megan. And as I said, we support shRNA, ORF, and CRISPR technologies. So in addition to providing these plasmid constructs, either as glycerol stalks, plasmids, and lentivirus from our pre-existing libraries, we also offer a bunch of custom services like custom cloning, similar to what Ralph's core does. We do cell engineering. We help with genetic screens and miscellaneous cloning stuff. We help PIs with their research and development aspects, optimizing their assays. We collaborate and consult and make sure that people are using the right resources to answer the right questions, grant preparation, and those standard things. So here are some metrics. We are a really small core, but we are core for 5 campuses in the state of Colorado. And we have supported research in more than 370 labs in the state of Colorado, and which has resulted in more than 270 publications. So I think despite the fact that we are small, we are mighty. So as I said, we do distribute a lot of individual shRNA or ORF constructs and have helped in knocking down, several hundred genes. And these are just some of the examples. I'm not going to waste time in going through this. But as I said, the real power and the real need for creating this core was wanting to do shRNA screens. So we've supported more than 17 shRNA screens. And they have been in cancer models, Down syndrome models, just understanding glucose biology. But Ralph gave some really good examples of how these screens can lead to drug development. In our case, a lot of these screens have led to clinical trials. So this is an example of an shRNA screen, which was done using a kinome library in context of Down syndrome. So the kinome shRNA library was transduced in 6 different cell lines, 3 cell lines that were created from patients, individuals who had Down syndrome, and then 3 lines from euploid donors. And this screen led to the identification of interferon pathway as being misregulated in individuals with Down syndrome. That led to identification of JAK as a targetable molecule. And less than 10 years later, we have several clinical trials going on where JAK inhibition has significantly improved lives of people with Down syndrome. And I feel really honored to be part of this journey. But this is just one example, and these are slides of different clinical trials that we have supported. And this is all shRNA, right? So as I said, 2013 is when CRISPR started, and although I want to say that adoption of CRISPR technology in Colorado has been relatively slow, we are getting there. So I was tasked with learning everything about CRISPR, not knowing anything about it. So as Ralph mentioned, CRISPR is a technology where you use an endonuclease, and a guide RNA directs this endonuclease to a specific region of the DNA, and it creates a double-stranded break, which gets non-specifically repaired, and thus creates a premature stop codon and hence gene knockout. So CRISPR is an amazingly powerful technology, so much so that it has completely taken over the field of functional genomics. As you gauge from Ralph and Guru's talk, it's all about CRISPR. And I'm sure it's easy enough that we can adopt it in context of cores and create cores around it, but we have to be mindful of where we are going. Okay, so we have an arrayed CRISPR library, so we do distribute reagents from that, but we do clone a lot more guide RNA. But again, the real interest is in doing genetic screens, similar to what Ralph was talking about, and cell engineering. So unlike Guru, I mostly generate cell line models rather than animal models. So I can knock out genes in cells, whether they're stem cells or cancer cell lines, or tag them, knock in mutations. So those are the kind of experiments that my core does. And then in 2014, I taught myself this. And then by 2017, we were able to publish the first CRISPR screen using the Gecko library that I amplified. And in the same year, we were able to publish another paper which used the knockout lines that I developed. So, and in those days, it was just me. Now I have at least one more person. But we have supported a lot of CRISPR screens, whether it is genome-wide libraries. And these libraries you can buy from @Gene, amplify them carefully, QC them with NGS, and provide them as pools of plasmid or virus. So we've done a bunch of those. We've also supported a screen which used the pre-made kinome CRISPR library. But I also want to talk to you about custom CRISPR libraries. So you give me a list of genes, we can design and create a specific, a very custom CRISPR library. One of the first libraries that we created is called CUDP, or CU Druggable Panel. This library has guides for about 1,200 genes that can be targeted by FDA-approved drug. So you do the screen using CUDP library, and you identify your hit, and there is a drug that you can test and move your experiments to clinical testing and animal models. Oh, so yesterday there was talk about development of an antibody for dogs in the curiosity section. So I did want to mention this, that we have designed, I want to say, first of its kind, whole genome canine library for designing or identifying drugs for our companion animals. So stay tuned, it's being tested. Okay, so we've done a lot of knockout. We've created a lot of cell lines with gene knockout. We've tagged a lot of cells, and we can do it quite efficiently. And the more you do it, the more you learn, and you can apply lessons learned from one project to the next project. So we are definitely getting better at utilizing this technology. So what next for my core? So my core provides bits and pieces for doing CRISPR screens or shRNA screen, where we can provide the library, the virus. We can talk to you about how the experiment should be designed, and then maybe even help with the downstream sequencing library prep. So the only section that we have not been doing is the actual experiment. But after talking to Ralph, I feel like maybe we should do that. So I'm going to try to establish that as a service, end-to-end CRISPR or screening services. The other, coming to ABRF, I realized the potential of spatial and single-cell transcriptomics. And you can combine these readouts with CRISPR screens, and as you can imagine, it would be a very powerful technology. It has some of its limitation based on how many cells you can analyze, and hence how many genes you can target with CRISPRs. But this is another assay or protocol that I'm trying to optimize. So when people or PIs in Colorado are ready to embrace, I can help them design and execute these experiments. We have still not started working with CRISPR-I and CRISPR-A and all the other Cas enzymes, so that is definitely in my to-do list. But how do we decide what technologies we add to our core? And that's based on surveys that we annually conduct to see what services our investigator want us to incorporate. And also there is a balance between what they want versus what is feasible to incorporate into a core. So once we take all of those metrics into account, we kind of add new services to the core. So that was my spiel about our journey and where we have been, how we got here. But there were a couple of things I do want you guys to consider, or people who have functional genomics core, and we might have similar opinion on these matters, or groups that are planning to start functional genomics core. We are a different kind of beast. We are not instrument cores where you buy a multi-million-dollar instrument, and because you have the instrument, people will come. We are dealing with a technology which has democratized functional genomics. People can do it in the backyard. So that creates a problem in itself. But I'm going to gradually go through my list. Also, so first point is moving target. This is a very rapidly evolving field. So you really want to think where you want to invest your time and money, what are the right technologies that you want to bring into your core. It's, as I said, it's it's not an instrument-based, it's a personal-based core. That means you need extremely highly skilled people. You cannot have people who just want to show up for a couple of hours and then be done. You want people who can multitask. You want people who can think outside the box. My core is so small, it's just 2 people. That means, we wash dishes and consult, do it all. And honestly, even if I had more people, I would want the same characteristics or the same skill set in everyone. How involved do you want to be? Do you want to be Ralph? Do you have the resources to do it all? Or if you are a small enterprise, it's okay to just provide reagents and say, we are here to help, but you do the experiment because you are the expert in your cell line, in your disease model, your drug. So that is another consideration that you want to keep in mind. And of course, it's not one size fits all. Even if you optimize all your CRISPR, all your protocols perfectly, every cell line behaves differently. The PCR that works in one cell line often fails in another. So every project is like reinventing your protocols. So that brings you back to the fact that you need people who are able to do that. And that brings me to the point that cores like ours, and generally all cores, we should have a dedicated R&D fund so we don't have to try and recuperate 100% of the time that you're billing, because you always need to figure out, okay, what next you are going to do? How can you make this protocol more efficient? Adding semi-automation helps because, when you have to do single-cell cloning, it's very painful to sit at the microscope for hours and scan for single-cell clones. And not do it once. You have to do it every few days over and over again. It's painful. So, some moderate semi-automation helps. Project management is great because a lot of these projects are very long. And cell engineering, it can take weeks to months. Single-cell cloning can take a really long time. Same thing with generating custom CRISPR libraries. So, if you have a system which kind of tells your client labs that where you are in the process, it helps them. It can be as simple as sending them an email every few weeks, but some sort of management would be good. And then internal and external competition. As I said, this has democratized our field. So I can do it, my core has been doing it for so well. I have edited more than 100 cell lines, all different cell lines, edited multiple genes, and I have learned from every project. And I have figured out how I can make my process efficient. But if there is a postdoc who's done a few projects in another lab, and you don't have to pay him, people will probably go to him or her. And there are competing cores, right? My core has been mostly focusing on editing cancer cells, which, by the way, are extremely painful compared to iPSCs, which only have 2 copies, because in cancer cells, you have way too many copies sometimes. But there are iPSC core, and the point is, if I can do iPSC, maybe I can do cancer cell, maybe I can do stem cell, maybe I can do neuronal cell. So, there is always internal competition that way. External competition, well, companies can probably do it better or faster. And cheaper also maybe because of the bulk of work that they do. And they're completely automated, so hands-free. So you always have to think about that. And the most important point that I want to make is find your professional tribe. If this is the field that you want to go in, you need people who are in the same boat and can guide you through the process. And I found Ralph and Guru and Genome Engineering Research Group through ABRF. So now I have people who are in the same boat, who are doing work that is similar to mine, who are struggling with the same kind of financial issues or competitions or things not working. Where do I go? How do I troubleshoot this? So I shoot an email to Ralph anytime I have issue with a screening, with a library or screening. I have Chandra and Jen and Guru for a lot of gene editing issues. So yeah, please come join GURG if this is what you want to do. And that is my talk. And I would love to thank Guru and Ralph for accepting to be part of this session. I want to thank ABRF for organizing such an amazing meeting. And now we're ready to take questions.
Great talk, everybody. Eric Chow from UCSF, and I just want to comment, there was this discussion about cloning libraries. I just want to mention that about a year ago, we published a new method to clone libraries that really reduced the bias and increased the uniformity. That might be of interest if you're trying to clone libraries. Found that it can actually kind of reduce the cell numbers you need for screens. But we've also used a protocol to clone the single guide CRISPR-I, CRISPR-A, and Brunello libraries, and just making it available through an MTA if people are interested. So if you're interested in taking a look, it's called, you can go to the website, just lgr.bio. But yeah, so it could be a good resource.
Thank you.
So Brian at ARC Institute, I was wondering if you start offering this as a service and then for groups, and there might be lots of projects that are more ad hoc, that are, the feasibility is not that clear. How do you structure like cost, or how do you just charge for time? Do you charge for time? Because this is often much more labor intensive than just like running a sequencing core or something like that, right?
Yeah, thank you for this question. Because we struggled with this in the early days. So we looked at our first couple of years of running the core, and 90% of our jobs were custom jobs, were unlike any other job we had done previously, even though they involved SH or CRISPR. And so that meant that our price estimates were all over the place. Our recoveries were low. So Sloan Kettering assigned an accountant to my group. And he knew nothing about gene editing biology, and we admittedly didn't know much about accounting. So we met in the middle, and one day he came in and he said, "Eureka, I've got it, Lego." And I said, "What do you mean Lego?" He said, "Well, listen, you can get a box of Lego and you could build a church or a fire truck or a steeple or any of these things, but it's the same parts. So we're going to take every reagent, every disposable, every task that you do in your core, and we're going to put a value on it based on what your aggregate salaries are, And then when you get a new job come in, you're just going to pull and populate those things. And that allows you to create line items. And the line items are important because as you work through a multi-month project, each month you can make a billing event, right? So you don't go from huge revenues to no revenues. So that was a really important. We have to publish our prices, and we do. But what you don't see on our published price list is the Excel sheet that's behind that. And we consider that a competitive advantage for our core because now for the last 6 years, we've recovered between 85 and 93% of all our costs. And that pays our salaries, and it's all of our reagents and the electricity and the water running. So we do have to generate revenues, but that's a very important part of putting a core together is figuring out what that price list is. Thank you for that question.
Yeah, thanks.
Thank you for some great talks. I really appreciate it. I want to echo what Ralph says. We do it by milestones, we do it by Legos, and it works really well. Creating those Legos and the fact that they tend to be dynamic can be a little annoying, but it works. So I strongly recommend that. But that's once you have a core going, right? That's once you have a service up and going. And I'm very glad that you pointed out that there are cores. We develop brand new monoclonal antibodies from scratch, so our projects can go on for 2 years. That there are cores exist that don't run on big boy toys. I mean, yes, you guys have some big boy toys in your labs, I know you do, but we don't. And we are human, we are human driven. And there is currently no funding mechanism, unlike S10 grants, to help bring a technology into a core if it doesn't have a big boy toy. This is not the environment to be telling the NIH that we need another granting thing. But at some point in time, I think it would be wonderful if minimally institutions understood that to bring some technologies into a core is not just a big piece of equipment. So thank you for bringing that up.
Yeah, that's a great point, and Molly made that during her talk, is if you walked into my lab or Molly's lab, you wouldn't know what we do, right? If you walk in and you see a fax area, you know. If you walk in, you see a HiSeq, you know. If you walk in, you see an animal imager, you know. If you walk in and you see microscopy, you know. Our lab, you don't know. So it's not dependent on that equipment, so you absolutely have to be good at what you do. And a lot of times investigators will come to me and say, well, why should we use your core? I can have my graduate student or my postdoc, do this CRISPR screen. And I said, well, that's great, let them do it. But there's going to be trial and error, okay? We take the trial and error out because we do this every single week and we make sure that it works out right the first time. And while we're doing that screen for you or facilitating that screen with your graduate student or postdoc, they could be doing something else that's very productive. And then in a couple weeks, we'll get back to you either with that cell line or with the results of that pooled screen. So yeah, it's very important to get on top of your game and stay on top of your game because it's not equipment dependent.
Thank you for bringing up the point that, we don't have a funding mechanism. And I have been complaining to anyone who would listen that we are a different kind of core. We need different kind of funding resources. I have been pointed to R50s, but they are not easy to get. I applied for R50 for 4 years before getting a fundable score this year. So yeah, but then, it doesn't really fund like people who don't have, who have undergraduate degree. And we do have a lot of people who've been in labs like yours for years and are a treasure trove of knowledge, but just because they don't have a degree, they don't qualify for that. And even if they would, it's just a really difficult mechanism to get money. So if you want to start an interest group, let me know. I've been complaining about this for years.
Lovely talk. Just a very quick question about, I'm an automation guy, I'm software automation. How much automation is involved in your skill set? And you mentioned Molly, that you need highly skilled people. But is there any comment about standardization and automation? How much can you automate?
So when I say automation, these are basically small instruments which can do the repetitive jobs. Like, we do a lot of plasmid prep. Instead of my assistant doing plasmid prep, we got a Kaya cube, and that does the plasmid prep. So when I say automation, it's not like robotics or anything. And then we've recently applied for an S10 to get a cell selector flex platform, which will automate the cell picking and single cell cloning aspect. So that will be a huge, it will save our time quite a bit. But honestly, like our cores like ours, we don't have really big instruments that we can buy. So there is, it is a 100% human core. Okay. Well, thank you everybody for sticking around. And as I said, we are recruiting people to ABRF's Genome Editing Research Group. And we would love for people to join us. Together we are stronger.
Thank you all.
Thank you.